As the brain ages, virtually every aspect of its structure and function undergoes transformation. Neuronal connectivity declines, oxidative stress accumulates, and energy metabolism begins to falter. Among these changes, the handling of glycogen, the brain's emergency energy reserve, is emerging as a subtle yet potentially powerful piece of the puzzle. Once thought to be relatively insignificant in adult brain metabolism, glycogen, primarily stored in astrocytes, has gained attention for its nuanced role in supporting cognition, sleep, and neuronal resilience. With advancing age, however, this role appears to shift. The question now arises, is there a glycogen switch in the aging brain? And if so, could it help explain the brain's increasing susceptibility to neurodegenerative diseases? In young, healthy brains, glycogen plays a dynamic and protective role. Astrocytes synthesize and degrade glycogen in response to neuronal demands, stress, and circadian rhythms. During periods of heightened cognitive activity or metabolic strain, glycogen is mobilized to support neurons via the astrocyte neuron lactate shuttle. This shuttling helps maintain synaptic transmission and memory consolidation. Sleep, particularly deep non-REM sleep, replenishes glycogen stores, suggesting that this system is tightly synchronized with rest and recovery processes essential for long-term brain health. As aging sets in, however, this system begins to show signs of dysregulation. Several studies in rodents and human tissue suggest that the capacity for glycogen synthesis and turnover diminishes with age. The enzymes responsible for glycogen cycling, such as glycogen synthase and glycogen phosphorylase, may become less responsive to signaling cues, leading to slower or impaired mobilization. Additionally, aging astrocytes often display signs of metabolic stress, mitochondrial dysfunction, and inflammatory activation, all factors that could interfere with efficient glycogen regulation. One possibility is that the glycogen switch in aging involves a shift from fast, flexible metabolic response to a more static and inefficient storage pattern. Instead of being quickly mobilized when needed, glycogen may begin to accumulate in less accessible or even pathological forms. Poorly branched glycogen, sometimes resembling polyglucosan bodies, has been observed more frequently in aged brains and in early stages of neurodegenerative diseases. These structures are harder to degrade, can crowd cellular space, and may interfere with normal astrocyte function and neuron glia signaling. In this context, glycogen no longer serves as a protective reservoir, it becomes a burden. This metabolic rigidity could have profound consequences. If aged astrocytes are less capable of supplying fuel to neurons during periods of stress, such as during hypoglycemia, sleep deprivation, or inflammation, then neuronal circuits become more vulnerable to damage. The hippocampus, central to memory formation, is especially sensitive to energy fluctuations. An impaired glycogen buffer in this region could contribute to age-related memory loss and pave the way for early Alzheimer's pathology. Adding another layer of complexity, aging is also associated with changes in insulin sensitivity, both peripherally and within the brain. Since insulin and IGF, one's signaling influence glucose uptake and glycogen metabolism in glial cells, the emerging picture is one of a systemic shift in how the brain accesses and stores energy. This could partly explain the link between metabolic disorders like type 2 diabetes and insulin resistance and elevated risk of cognitive decline and dementia in older adults. Yet not all the news is grim. Some studies suggest that lifestyle interventions known to preserve cognitive function in aging, such as intermittent fasting, aerobic exercise, and deep sleep, also support healthier glycogen cycling. These behaviors may help maintain enzyme responsiveness, reduce astrocytic inflammation, and preserve the rhythmic fluctuations in glycogen that underpin resilience. If the glycogen switch of aging is real, it might also be modifiable. Furthermore, tracking these glycogen shifts could open new diagnostic or therapeutic possibilities. Advanced imaging techniques like carbon-13 magnetic resonance spectroscopy, 13C, MRS, are beginning to offer glimpses into in vivo glycogen dynamics. If age-related impairments in glycogen storage or mobilization can be detected early, they may serve as warning signs for accelerated cognitive aging. Moreover, pharmacological tools that target glycogen metabolism, such as modulators of glycogen phosphorylase, might one day be deployed to restore metabolic flexibility and improve brain resilience. In conclusion, the concept of a glycogen switch in the aging brain offers a compelling framework for understanding how subtle metabolic changes can have outsized impacts on cognitive health. Glycogen, far from being a passive or redundant energy source, appears to operate as a tightly regulated buffer system, one that, when disrupted, could increase vulnerability to neurodegeneration. By exploring how this system evolves with age and how it interacts with broader metabolic networks, we may unlock new strategies to preserve brain function deep into old age. The glycogen switch, then, is not just a biological curiosity, it may be a keystone in the bridge between aging and cognitive decline.